Hello there, and welcome back to Bedtime Science. I am really glad you are here tonight. Somewhere on the surface of the body, a cell has noticed that something is different. The change may be extremely small, a microscopic crack in the skin, a virus entering one cell in the lining of the nose, a bacterium moving past mucus into a place where bacteria are not usually allowed to remain. Nothing about the moment needs to be dramatic. There may be no pain yet, no fever, and no conscious awareness that anything has happened. But molecules have changed position. A membrane has been disturbed. A receptor has bound a pattern. A cell has begun sending a message. That message will travel through tissue, blood, and lymph. It may summon cells that were produced in bone marrow days or weeks earlier. It may reach a lymph node where a rare T cell, one among millions, happens to carry a receptor capable of recognizing a fragment of the intruder. It may lead a B cell to divide, mutate, compete, and eventually release antibodies shaped by that encounter. Or the disturbance may be resolved before the adaptive immune system is needed at all. The immune system is often described as an army defending a border. The image is useful and incomplete. An army mainly waits for enemies. The immune system also removes dead cells, repairs tissue, shapes developing organs, cooperates with microorganisms, restrains inflammation, watches for cancer, and decides when not to attack. Its central problem is not simply recognizing what belongs to the body and destroying everything else. Food is foreign. Pollen is foreign. Most microorganisms living peacefully in the intestine are genetically foreign. A transplanted organ may be healthy and still recognized as foreign. A damaged cell belongs to the body and may still need to be removed. A cancer cell began as self and can become dangerous while retaining many signs of self. The immune system therefore responds not only to identity, but to location, context, damage, repetition, and the kind of molecular pattern being detected. It asks several questions at once. What is this? Where is it? Is tissue being harmed? Has this been seen before? Which kind of response would remove the threat with the least injury to the host? And when should the response stop? Tonight, we will follow one disturbance from the body's surface inward. We will begin with barriers that prevent most encounters from becoming infections. We will watch inflammation open local blood vessels and guide neutrophils into tissue. We will meet macrophages that lived in organs long before the current infection began, and dendritic cells that carry evidence toward a lymph node. Inside that node, we will see how T cells and B cells search through possibilities created by rearranging their own DNA. We will follow antibodies as they improve, memory cells as they remain, and regulatory cells as they prevent defense from becoming self-destruction. Then the story will widen. Vaccines will turn memory into medicine. Allergy will reveal what happens when harmless material is assigned the wrong meaning. Autoimmune disease will show how tolerance can fail. Cancer immunotherapy will show how an existing response can be released from inhibition, redirected, or expanded outside the body and returned. Finally, we will come back to the quiet tissue where the first signal began. By then, the invader may be gone. The response itself will need to disappear. The immune system's success will be measured partly by what no longer happens. No spreading infection, no continuing fever, no endless inflammation, only repaired tissue, altered memory, and a body returning to ordinary life. The first defense is the surface. Skin appears simple because most of its work is silent. Its outer layer, the stratum corneum, consists largely of flattened cells filled with keratin and embedded in lipids. These cells are no longer living in the usual sense. They form a flexible wall that resists water loss, chemicals, and microbial entry. The wall is renewed continuously from below. Keratinocytes divide in deeper layers, move outward, change their proteins and lipids, and eventually become part of the barrier that will be shed. The skin is not sterile. Bacteria, fungi, viruses, and microscopic arthropods occupy its folds, follicles, and oily or dry regions. Different communities prefer the moist environment of an armpit, the oily environment of the face, or the drier skin of the forearm. Many residents cause no harm and can make invasion harder by using nutrients, occupying attachment sites, producing inhibitory chemicals, and influencing local immunity. The relationship is not one permanent peace treaty. A harmless resident on the surface may cause disease if it enters deeper tissue. A community can change when moisture, temperature, antibiotics, inflammation, or skin damage changes its environment. The immune system must therefore tolerate presence in one place and respond to the same organism in another. Location carries meaning. The surface also contains chemical defenses. Its acidity restricts some microbes. Sebum contains lipids that can be modified into antimicrobial molecules. Keratinocytes produce peptides that disrupt microbial membranes and signaling molecules that alert nearby cells. Tight junctions reduce passage between cells. Hair follicles contain immune cells positioned near one of the natural openings through the barrier. Inside the nose, mouth, lungs, digestive tract, and reproductive system, the body cannot use a dry wall. These surfaces must exchange gases, absorb nutrients, sense the environment, or permit reproduction. They are covered by mucous membranes. Mucus is not simply sticky water. Large glycoproteins called mucins form a hydrated network. The network traps particles and microorganisms while allowing selected molecules to diffuse. Antimicrobial peptides, enzymes, and antibodies enter the layer. In the airways, cilia beat in coordinated waves and move mucus upward toward the throat. The motion is called the mucociliary escalator. Particles inhaled during the day can be carried away without the adaptive immune system ever learning their exact shapes. Coughing and sneezing accelerate removal. Tears wash the eyes. Saliva dilutes and carries material from the mouth. Urine flow reduces microbial attachment in the urinary tract. Stomach acid destroys many organisms swallowed with food, although some survive by being protected inside food, producing acid resistance, or moving quickly into less acidic regions. The intestine presents the most elaborate barrier problem. It contains an enormous microbial community close to a surface only 1 cell thick. The epithelial cells must absorb nutrients while preventing microbes and toxins from entering tissue. Mucus separates many organisms from the epithelium. Tight junctions connect neighboring cells. Specialized cells produce antimicrobial peptides. Secretory immunoglobulin A coats microorganisms and limits their contact without necessarily triggering destructive inflammation. Immune cells beneath the epithelium sample what passes nearby. Most food and resident microbial material is met with restraint. A strong inflammatory response to every meal would make digestion impossible. The immune system at a barrier is therefore not inactive. It is actively tolerant. It maintains distance, limits growth, repairs damage, and chooses responses that preserve the surface. Now imagine a splinter entering the skin. The wood carries molecules from the environment and may carry bacteria. The mechanical injury breaks cells and small blood vessels. Material that normally remains inside cells spills into the extracellular space. Microbial molecules appear in a compartment where they do not belong. Resident cells detect both kinds of information. The body has not yet identified one exact bacterial strain. It has recognized patterns of invasion and damage. The first message begins. Cells detect danger through families of receptors that appeared long before antibodies and T-cell receptors evolved. These pattern recognition receptors do not search through every possible molecular shape. They recognize recurring features associated with microbes, cell damage, or abnormal location. A bacterial lipopolysaccharide, a piece of flagellin, Double-stranded RNA in a cellular compartment where it can indicate viral replication. DNA lacking the chemical patterns common in human nuclei. Crystals, extracellular ATP, damaged mitochondria, or proteins released from injured cells. The microbial patterns are often called pathogen-associated molecular patterns. Signals from injured tissue are often called damage-associated molecular patterns. The categories overlap and remain more complex than their names suggest. A harmless microbe can carry a microbial pattern. A sterile injury can trigger inflammation without any infection. A receptor provides evidence, not a complete verdict. One of the most famous receptor families is the toll-like receptor family. The name came from a fruit fly. In the 1980s, developmental biologists studied mutations that changed how a Drosophila embryo established its body axis. One gene produced such a striking developmental pattern that Christian Nusslein-Volhard used the German expression toll, meaning something like amazing or fantastic. The toll protein seemed at first to belong to embryonic development. Then Jules Hoffman and colleagues found that adult flies also use toll signaling to defend against fungal infection. Flies did not have the vertebrate adaptive immune system, yet they could activate targeted antimicrobial programs through an inherited receptor pathway. The discovery connected developmental biology with immunity. At nearly the same time, Bruce Beutler was studying why bacterial lipopolysaccharide could trigger a dangerous systemic inflammatory response in mammals. Some mouse strains were unusually resistant to the molecule. The responsible mutation led to a mammalian relative of toll, now called toll-like receptor 4. A receptor had linked one conserved microbial structure with inflammatory signaling. These discoveries helped establish a principle proposed clearly by immunologist Charles Janeway. Adaptive immunity does not activate safely through antigen recognition alone. It needs information from the older innate system about the context in which the antigen was encountered. The antigen provides specificity. The innate signal provides meaning. Toll-like receptors are only one family. NOD-like receptors survey the cytoplasm and can assemble inflammasomes. RIG1-like receptors detect forms of viral RNA. C-type lectin receptors recognize carbohydrate structures common on fungi and other microbes. Cytosolic DNA sensors can activate interferon pathways. The sensors are distributed according to where their ligands would be suspicious. A receptor facing the cell surface can detect extracellular material. A receptor inside an endosome can inspect nucleic acids taken up from outside. A cytosolic receptor can detect material that has reached the cell interior. Compartment is part of recognition. When pattern recognition pathways activate, the cell changes gene expression. Transcription factors enter the nucleus. Cytokines and chemokines are produced. Antimicrobial programs begin. Interferons warn neighboring cells that a virus may be present. Endothelial cells lining nearby blood vessels change their surfaces. The tissue becomes inflammatory. Inflammation is often described by heat, redness, swelling, pain, and loss of function. These signs were recognized in ancient medicine long before the cells and molecules were known. They are not 5 separate accidents. They arise from coordinated changes. Small vessels widen, increasing local blood flow and producing warmth and redness. The endothelial barrier becomes more permeable, allowing fluid and plasma proteins to enter tissue. The additional fluid produces swelling. Chemical mediators sensitize nerve endings, making touch or movement painful. Pain discourages use of the injured area. Reduced function can protect tissue while repair begins. The same response that helps contain infection can become harmful if it is too strong, too widespread, or too prolonged. Inflammation is not a synonym for disease. It is a program whose value depends on scale and timing. Mast cells are among the resident cells capable of starting this program rapidly. They sit near blood vessels and barrier surfaces, filled with granules containing histamine and other mediators. When activated by injury, microbial signals, complement fragments, or antibodies in an allergic response, they release these substances within seconds or minutes. Histamine changes vessel diameter and permeability. Other mediators attract leukocytes, alter smooth muscle, and sustain the response. Mast cells evolved in contexts that include defense against parasites, toxins, and venoms, although their full physiological roles remain broader than one explanation. The same machinery produces the immediate symptoms of allergy when harmless material is assigned dangerous meaning. The blood vessel now becomes an active gate. Circulating immune cells do not simply leak through a hole. Inflammatory cytokines cause endothelial cells to display adhesion molecules. White blood cells passing in the bloodstream make weak contacts, slow, and roll along the vessel wall. Chemokines presented on the endothelial surface activate stronger adhesion. The cells stop. They flatten and squeeze between or through endothelial cells into tissue. This is extravasation. The cells then follow chemical gradients toward the source. The process resembles navigation by smell, though the signals are concentrations of molecules detected by receptors. A cell repeatedly compares the stimulation across its surface and reorganizes its internal skeleton to move. Among the first large waves of arriving cells are neutrophils. Neutrophils are the most abundant leukocytes in ordinary human blood. They are produced continuously in bone marrow and maintained in large reserve pools that can be released during infection or injury. Their exact lifespan in human circulation has been debated for years. Older estimates suggested only several hours. Labeling and modeling studies have produced longer values, extending into days under some assumptions. The important feature is not one fixed number. Neutrophils are relatively short-lived, rapidly deployed cells built for intense local activity. They follow chemokines, complement fragments, bacterial products, and signals released by damaged tissue. At the splinter, they encounter bacteria attempting to divide. A neutrophil can wrap its membrane around a bacterium and enclose it inside a phagosome. Granules fuse with the compartment. Enzymes, antimicrobial peptides, and reactive oxygen species attack the captured organism. The chemical environment is destructive by design. The neutrophil can also release granule contents outside the cell. It can extrude networks of chromatin covered with antimicrobial proteins, called neutrophil extracellular traps. These nets can immobilize microbes. They can also damage tissue, promote clotting, and contribute to inflammatory disease when produced excessively or cleared poorly. The strongest weapons are never free. Pus is formed partly from dead and dying neutrophils, tissue fluid, microbes, and cellular debris. Its presence reflects the density of the response, not one particular pathogen. Most neutrophils eventually undergo a controlled form of death. Macrophages engulf them before their contents spill widely. The disposal of neutrophils helps change the tissue from an inflammatory state toward resolution. The response is already preparing for its own ending. Macrophages were present before the neutrophils arrived. Their name means large eaters, but eating is only one part of their work. They sense, engulf, signal, remodel, recycle, and repair. The old textbook pathway described a monocyte leaving blood and becoming a tissue macrophage. That pathway is real during inflammation and in several tissues. It is not the origin of every resident macrophage. Many macrophage populations seed tissues during embryonic development, before definitive adult blood formation is established. They can remain for years and renew themselves locally. Microglia in the brain are the clearest example. Kupffer cells in the liver, many alveolar macrophages in the lung, and other resident macrophage populations also have developmental histories that cannot be reduced to circulating monocytes. Langerhans cells in the epidermis are long-lived, embryonically seeded antigen-presenting cells with features connecting the macrophage and dendritic cell families. Inflammation can later add monocyte-derived cells, and the balance varies by organ, age, and injury. The tissue teaches each population a local identity. Long before anyone knew that tissues contained macrophage lineages with different embryonic origins, one biologist watched mobile cells gather around a thorn. In 1882, Ilya Mechnikov was working in Messina, Italy, studying transparent starfish larvae. The larvae had no blood vessels like those of a mammal and no adaptive immune system. Their simplicity made movement inside the living animal easier to observe. Mechnikov noticed wandering cells in the clear body. He wondered whether they did more than transport nutrients or shape developing tissue. To test the idea, he inserted tiny thorns from a tree into the larvae. By the following morning, mobile cells had surrounded the foreign material. The observation was small enough to fit beneath a microscope. Its implication was much larger. Perhaps inflammation was not only damage caused by invading organisms. Perhaps cells actively moved toward danger and engulfed what did not belong in the tissue. Mechnikov called the process phagocytosis, from Greek words associated with eating. He later observed related behavior in other animals and argued that phagocytes formed a fundamental cellular defense against infection. The idea was controversial. Other researchers emphasized soluble substances in blood, the line of investigation that would lead toward antibodies and complement. The dispute was sometimes framed as cellular immunity against humoral immunity. The body did not choose between them. Phagocytes recognized targets directly and also through antibodies and complement deposited on their surfaces. Cells and soluble molecules cooperate. In 1908, Mechnikov shared the Nobel Prize in Physiology or Medicine with Paul Ehrlich, whose work helped establish the specificity of antibody-like defenses. Their approaches had appeared opposed and became parts of one immune system. The starfish experiment also contained a clue that modern immunology would recover repeatedly. Cells gathered around a sterile thorn even when no microbe was present. Tissue damage itself carried information. The splinter in this story therefore resembles the experiment that helped make cellular immunity visible. The cells around it are not repeating one ancient program unchanged. They are integrating microbial patterns, broken tissue, complement, cytokines, and the local needs of skin. But the first question remains recognizable. What brings a wandering cell toward a place where the boundary has been broken? A macrophage in the liver encounters blood arriving from the intestine and participates in clearing aged blood cells and managing iron. An alveolar macrophage lives on a thin gas exchange surface where unnecessary inflammation would interfere with breathing. Microglia extend processes through neural tissue, responding to injury while also shaping synapses and development. Macrophages near the splinter receive a different set of instructions. They detect bacterial and damage signals. They release tumor necrosis factor, interleukin-1, interleukin-6, chemokines, lipid mediators, and other molecules. The names can make cytokines sound like individual commands. Most act as parts of networks. The same molecule can have different effects depending on concentration, timing, receptor, tissue, and the other signals present. Tumor necrosis factor can activate endothelial cells and help contain local infection. In an uncontrolled systemic response, the same pathway can contribute to shock and organ injury. Interleukin-1 can promote fever and leukocyte recruitment. It can also contribute to chronic inflammatory disease. Immune signaling is contextual because the body cannot use one molecule for only one purpose. Macrophages engulf bacteria, damaged tissue, and dying neutrophils. Recognition can occur directly through receptors or indirectly after the target has been coated with molecules that mark it for uptake. This coating is called opsonization. Antibodies can act as opsonants. Complement proteins can do the same before antibodies exist. The complement system consists of plasma proteins produced mainly by the liver and circulating in inactive forms. A small initiating event can activate one protein, which activates several others, creating a cascade. There are three major routes into the cascade. The classical pathway can begin when antibodies bind a target, although some related molecules can initiate it without antibody. The lectin pathway begins when pattern recognition molecules bind characteristic microbial carbohydrates. The alternative pathway uses the continuous low-level activation of complement components and becomes amplified on surfaces that do not carry the regulators protecting human cells. The pathways converge at complement component C3. Cleavage produces fragments with different roles. C3b attaches to nearby surfaces and marks them for phagocytosis. Smaller fragments help recruit and activate inflammatory cells. Later components can assemble a membrane attack complex that inserts pores into selected microbial membranes, especially certain gram-negative bacteria. Complement also helps remove immune complexes and dead material. The system is powerful because it amplifies rapidly. It is dangerous for the same reason. Human cells display regulatory proteins that interrupt the cascade. Plasma inhibitors restrain activated enzymes. Without these controls, complement can injure blood cells, kidneys, vessels, and other tissues. Inherited deficiencies reveal the specialization. Deficiency in early classical components can impair clearance and increase susceptibility to immune complex disease. Deficiency in terminal components creates a characteristic vulnerability to Neisseria species. The pattern of infection can reveal which part of the cascade is missing. Clotting and immunity also communicate. A damaged vessel needs to stop bleeding. Platelets adhere, change shape, and recruit more platelets. Coagulation produces fibrin, creating a mesh that stabilizes the plug. The mesh can also help confine microbes locally. Immune cells activate coagulation, and coagulation products alter inflammation. This cooperation is useful at a small wound. When activated throughout the circulation during severe infection, it can produce microvascular clots and consume the components needed to prevent bleeding elsewhere. Sepsis is not simply an infection that became large. It is life-threatening organ dysfunction caused by a dysregulated host response to infection. Microbes, inflammation, vascular leakage, altered coagulation, metabolic changes, and impaired tissue oxygen delivery interact. The immune system can become simultaneously overactive and ineffective. Some pathways release overwhelming inflammatory signals early. Others enter exhaustion or immunosuppression later. The old image of a uniform cytokine storm captures only part of the process. Local inflammation remains different. At the splinter, the vessel changes are limited. Neutrophils and monocytes enter where they are needed. Complement deposits near the microbes. Fibrin helps close damaged vessels. The body may also change systemically in a controlled way. Fever is one example. Cytokines produced during infection influence the hypothalamus through neural and humoral pathways. Prostaglandin E2 raises the regulated temperature set point. The body then behaves as though its current temperature is too low. Skin vessels constrict. Muscles may shiver. A person feels cold while body temperature rises. Fever can slow some pathogens and alter immune performance. It also increases metabolic demand and can become harmful at extremes or in vulnerable people. Fever is not the body accidentally overheating in the same way as heat stroke. It is a regulated change in set point. When the set point returns downward, vessels dilate, and sweating helps release heat. Sickness behavior accompanies many immune responses. fatigue, reduced appetite, less interest in activity or social interaction, more sleepiness. These changes are not merely signs that pathogens have stolen energy. Cytokines communicate with the nervous system and alter behavior. Rest can reduce exposure, redirect energy, and support aspects of defense and repair. The response is adaptive in moderation and distressing or harmful when prolonged. The nervous and immune systems are not separate departments exchanging occasional messages. Nerves innervate lymphoid organs and tissues. Immune mediators affect neurons and glia. Hormones and circadian clocks change immune cell trafficking and responsiveness. The experience of being ill emerges from communication across the whole organism. At the wound, macrophages begin changing state as the threat declines. They engulf apoptotic neutrophils. This process can reduce inflammatory signaling and promote mediators associated with resolution. Resolution is not passive decay. Specialized lipid mediators help stop further neutrophil recruitment, promote clearance, and support tissue restoration. Fibroblasts produce extracellular matrix. Endothelial cells repair vessels. Epithelial cells migrate and divide to close the gap. Macrophages release growth factors, remodel matrix, and help coordinate these processes. Too little repair leaves a wound open. Too much produces fibrosis and scar. The immune system must remove a threat without replacing functional tissue with excessive connective material. The local response may already succeed, but some antigen has been taken up by a dendritic cell. That cell is preparing to leave the tissue. The next stage will occur in a lymph node, where specificity is created not by recognizing a broad microbial pattern, but by finding one rare lymphocyte among a vast repertoire. Dendritic cells were difficult to understand because they were rare and did not fit comfortably into the categories used to describe other white blood cells. In 1973, Ralph Steinman and Zanvil Cohn reported an unusual population in the mouse spleen. The cells carried long branching processes. Steinman named them dendritic cells, from the Greek word for tree. At first, many immunologists doubted that a rare cell type could have the central role Steinman proposed. Macrophages were already known to engulf material and display antigens. Why would another antigen-presenting cell be needed? Steinman continued isolating the cells, improving methods, and showing that they were exceptionally effective at activating naive T cells. The distinction mattered. A macrophage in infected tissue can destroy microbes and stimulate experienced effector cells. A dendritic cell can carry information from that tissue into a lymphoid organ and initiate a response in T cells that have never encountered the antigen before. It links place with specificity. Ralph Steinman died from pancreatic cancer on September 30, 2011. 3 days later, the Nobel Committee announced that he would receive half of the Nobel Prize in Physiology or Medicine for discovering dendritic cells and their role in adaptive immunity. The committee had not known of his death when it made the decision. Nobel Prizes are normally not awarded posthumously, but the award remained because the decision had been made while he was alive. The unusual timing became part of the public story. The scientific story had taken nearly 4 decades. In the skin near the splinter, a dendritic cell takes up bacterial proteins and damaged material. The proteins are broken into peptide fragments inside intracellular compartments. Some fragments are loaded onto major histocompatibility complex molecules. MHC class II molecules display peptides mainly to CD4 helper T cells. MHC class I molecules, expressed on nearly all nucleated cells, display peptides from proteins generated inside the cell to CD8 T cells. Dendritic cells can also perform cross-presentation, placing material taken up from outside onto MHC class I and thereby activating cytotoxic T cells against viruses or tumors that may not infect the dendritic cell directly. Peptide presentation is only one part of activation. A naive T cell needs co-stimulatory signals. A dendritic cell that encounters inflammatory or microbial cues matures. It increases MHC and co-stimulatory molecules, changes chemokine receptors, and begins migrating through lymphatic vessels. A dendritic cell carrying ordinary self-material from uninjured tissue may instead promote tolerance or fail to activate a naive T cell fully. The same peptide can therefore be interpreted differently depending on the state of the presenting cell. Context reaches the lymph node on the surface of another cell. The lymphatic system begins in blind-ended vessels within tissues. Fluid that leaves blood capillaries does not all return directly to them. Some enters lymphatic capillaries, carrying proteins, particles, immune cells, and antigens. The vessels converge and pass through lymph nodes before lymph eventually returns to the bloodstream. A lymph node is not a passive filter like cloth. It is an organized meeting place. Its architecture increases the chance that rare cells will find the information meant for them. Incoming lymph enters beneath the capsule and moves through sinuses. Macrophages capture particles and immune complexes. Dendritic cells migrate into T cell zones. B cells occupy follicles. Specialized stromal cells create a network along which lymphocytes move and provide chemokines that keep different populations in the correct neighborhoods. High endothelial venules allow naive lymphocytes to leave blood and enter the node. A lymphocyte may spend hours moving through this environment, touching many antigen-presenting cells, and then depart if no receptor matches. The movement looks random at small scales and is guided statistically by structure and chemical signals. Millions of cells circulate repeatedly through blood, lymph, spleen, and nodes. The immune system solves rarity by continuous traffic. The spleen performs a related form of surveillance for the bloodstream. It sits in the upper left abdomen and receives a large blood flow. Its red pulp filters aging or damaged red blood cells and recycles iron. Its white pulp organizes lymphocytes around blood vessels and initiates responses to blood-borne antigens. Marginal zone B cells and macrophages can respond rapidly to encapsulated bacteria passing through blood. This is why people without a functioning spleen face particular risk from organisms such as Streptococcus pneumoniae, Haemophilus influenza, and Neisseria meningitidis. Vaccination and, in selected situations, preventive antibiotics reduce that risk. The spleen is not merely a large lymph node. A lymph node samples lymph arriving from a region of tissue. The spleen samples blood. The tonsils and adenoids sample material entering through the mouth and nose. Peyer's patches sample the intestine. The immune system places organized structures at routes where information is likely to arrive. A swollen lymph node reflects activity, not necessarily a dangerous condition. Lymphocytes divide. Blood flow changes. Macrophages collect material. Stromal networks expand. A node draining a throat infection may become tender because its capsule stretches and local inflammation activates nerves. Nodes can also enlarge during autoimmune disease or cancer, and persistent or unusual swelling deserves medical evaluation. A naive T cell receptor does not bind free antigen in the way an antibody can. It recognizes a composite surface made from one peptide held by one MHC molecule. The receptor touches both. This is MHC restriction. A T cell is trained to recognize foreign or altered peptides only when presented by the body's own MHC framework. Human MHC genes are called human leukocyte antigen genes, or HLA. They are among the most variable genes in the human population. Different HLA molecules bind different peptide sets. This diversity helps a population survive varied pathogens because one pathogen is unlikely to evade presentation in everyone. The same diversity complicates organ transplantation. A donor's HLA molecules can appear profoundly foreign to a recipient's T cells. Matching reduces risk, but rarely makes two unrelated immune systems identical. The dendritic cell displays thousands of peptide MHC complexes. T cells pass over its branching surface. Most contacts end quickly. One T cell receptor binds sufficiently well to a bacterial peptide displayed by the correct MHC molecule. Adhesion stabilizes the interaction. Co-stimulatory molecules engage. Cytokines influence the developmental direction. Signals accumulate across time rather than functioning as one instantaneous switch. The T cell changes gene expression, metabolism, and behavior. It begins producing interleukin-2 and increasing the high-affinity receptor for that growth signal. It divides. The first division is followed by another and another. One rare cell becomes a clone of thousands or millions sharing the same receptor. This is clonal selection. The antigen did not instruct the cell to manufacture a matching receptor. The receptor existed before exposure. The antigen selected the cell that already carried it. That principle was developed through the work of Niels Jerne, David Talmage, Frank McFarlane Burnett, and others in the mid-20th century. It solved one puzzle and exposed another. How could the body create enough different receptors before knowing which pathogens would appear? The answer required a change in the idea that every cell in one body keeps its genes arranged in exactly the same way. Developing lymphocytes deliberately rearranged their DNA. For much of the 20th century, antibody diversity appeared to require an impossible number of genes. If every antibody specificity were encoded as one complete inherited gene, the genome would need to devote an enormous portion of itself to immune receptors alone. Another proposal suggested that one small set of genes could somehow be altered after exposure to antigen. The central question was whether diversity was inherited in complete form or assembled during the life of each lymphocyte. Susumu Tonegawa approached the problem using the new tools of molecular biology. He compared DNA from embryonic cells, which had not developed into antibody-producing lymphocytes, with DNA from mature B-cell tumors producing one antibody. If antibody genes remained arranged identically in every cell, corresponding pieces of DNA should appear in the same configuration. They did not. Segments that were separated in embryonic DNA had been brought together in the B cell. The genome had been rearranged somatically. Tonegawa received the 1987 Nobel Prize for discovering the genetic principle that generates antibody diversity. The result changed a basic assumption. Cells usually preserve genome organization carefully. Developing B and T lymphocytes create controlled breaks, delete intervening DNA, and join selected segments to build antigen receptor genes that did not exist in that form in the fertilized egg. For immunoglobulin heavy chains and some T cell receptor chains, variable, diversity, and joining segments contribute. For other chains, variable and joining segments are combined. The process is called VDJ recombination. The RAG1 and RAG2 proteins recognize recombination signal sequences beside the segments and bring selected pieces together. DNA is cut. Repair machinery joins the ends. The joining is intentionally imprecise. Nucleotides can be removed. Additional nucleotides can be added by terminal deoxynucleotidyl transferase. The junctions form the most variable part of many receptors, often positioned where the receptor touches antigen. Combinatorial choice among gene segments creates diversity. Imprecise joining creates more. Pairing two independently rearranged receptor chains multiplies it again. The theoretical number of possible receptors is immense, far exceeding the number of lymphocytes present in one person at one time. The body samples a fraction of the possible repertoire. Random generation creates useful receptors. It also creates receptors that cannot function or that recognize the body dangerously well. Development must therefore include testing. B cells undergo much of this testing in bone marrow. T cells undergo it in the thymus. The thymus lies behind the upper sternum. It is relatively large and active in childhood, then gradually becomes smaller and more fatty with age, although functional tissue can persist. Immature T cell precursors arrive from bone marrow without a completed receptor. They rearrange receptor genes and move through distinct thymic regions. The first major test is positive selection. A developing T cell must recognize the body's own MHC molecules weakly enough to use them later. A receptor that cannot interact with self-MHC at all will be unable to recognize peptide MHC on infected cells. That cell dies by neglect. The surviving cells face negative selection. If a receptor binds self-peptide MHC too strongly, the cell may be deleted or diverted into a regulatory lineage. The thymus must display self-proteins from many organs that are not physically present there. Medullary thymic epithelial cells use transcriptional regulators, including AIRE, to express proteins normally associated with pancreas, skin, eye, and other tissues. Fragments are presented to developing T cells. The thymus performs a partial preview of the body. The preview is not complete. Binding thresholds are imperfect. Some self-reactive cells escape. Peripheral tolerance will be needed throughout life. The thymus also determines whether many cells become CD4 or CD8 T cells. Cells whose receptors interact with MHC class II generally enter the CD4 lineage. Cells recognizing MHC class I generally enter the CD8 lineage. The labels correspond to co-receptor proteins that stabilize the interaction and help signal. CD8 cytotoxic T cells inspect peptides produced inside cells. This makes them suited to detecting viral proteins or abnormal tumor proteins displayed on MHC class I. Once activated, they kill selected targets by releasing perforin and granzymes or engaging death receptors. Perforin helps granzymes reach the target cell interior. Granzymes activate controlled cell death pathways. The target dismantles itself rather than simply bursting and releasing viral contents widely. The cytotoxic T cell can detach and move to another target. CD4 T cells coordinate rather than perform one single helper function. Depending on cytokines, antigen, tissue, and transcriptional programs, they can become several functional states. T helper 1 cells support macrophage activation and responses to intracellular microbes. T helper 2 cells support defense against helminths and contribute to allergy. T helper 17 cells support barrier defense against extracellular bacteria and fungi and can contribute to autoimmune inflammation. T follicular helper cells enter B cell follicles and guide antibody maturation. The categories are useful without being absolute boxes. T cells can show mixed states and change over time. Another CD4 lineage prevents response from becoming destruction. Regulatory T cells express the transcription factor FOXP3 and suppress excessive or self-reactive immunity through several mechanisms. They consume growth signals such as interleukin-2, produce inhibitory cytokines, alter antigen-presenting cells, and restrain neighboring lymphocytes. Their discovery resolved a long argument. Experiments early in the 20th century suggested that removing the thymus from newborn animals caused immune abnormalities, but suppressor T cells later became controversial because the populations were poorly defined. In 1995, Shimon Sakaguchi identified a population of CD4 T cells carrying high levels of CD25 that prevented autoimmune disease when transferred into mice. Remove those cells, and self-reactive responses emerged. Restore them, and tolerance returned. The cells were not merely weak responders. They actively maintained restraint. A second path came from a mutant mouse called scurfy. Affected male mice developed severe, widespread inflammatory disease. Mary Brunko, Fred Ramsdell, and colleagues identified the responsible gene, later named FOXP3. Mutations in the human gene cause IPEX syndrome, a severe early-onset disorder involving autoimmunity, intestinal disease, allergy, and endocrine damage. Sakaguchi and others then connected FOXP3 with the regulatory T-cell lineage. The cells and the gene became parts of one mechanism. In 2025, the Nobel Prize in Physiology or Medicine was awarded to Mary Brunko, Fred Ramsdell, and Shimon Sakaguchi for discoveries concerning peripheral immune tolerance. The timing gave modern immunology a clear statement. The system's sophistication is not measured only by how many targets it can recognize. It is measured by how selectively it can refuse to respond. The activated T-cell in our lymph node has passed through these layers of selection. Its receptor fits the bacterial peptide. Its co-stimulation confirms inflammatory context. Its clone expands. Some descendants remain in the node and help B cells. Others enter lymph and then blood, following adhesion molecules and chemokines back toward inflamed tissue. The adaptive response is now becoming large enough to change the local battle. A B cell carrying a matching receptor has also encountered antigen. Its path will produce antibodies, but only after another round of selection that occurs inside a temporary structure called a germinal center. A B cell receptor is an antibody anchored in the cell membrane. Unlike a T cell receptor, it can bind intact molecular shapes. A protein surface, a carbohydrate arrangement, a repeating structure on a viral particle. When antigen binds and cross-links the receptor, the B cell internalizes the material, breaks proteins into peptides, and displays them on MHC class II. The B cell can now present evidence to a T follicular helper cell that recognizes a peptide from the same antigen. This linked recognition provides a safeguard. The B cell recognizes the intact object. The T cell recognizes a processed fragment. Both must agree that the material belongs to the same encounter. Contact between CD40 on the B cell and CD40 ligand on the T cell supplies an essential signal. Cytokines provide additional instructions. Some activated B cells quickly become short-lived plasma cells outside follicles. They release early antibody, often immunoglobulin M. Immunoglobulin M can form pentamers, giving many binding sites and making it effective at agglutination and complement activation even before each binding site has extremely high affinity. Other B cells enter the follicle and create a germinal center. The quiet architecture of the lymph node changes visibly under a microscope. B cells divide at extraordinary speed in one region. An enzyme called activation-induced cytidine deaminase introduces mutations into the variable regions of antibody genes. Mutation usually threatens function. Here it creates a local evolutionary experiment. Each daughter cell may carry an antibody receptor with slightly altered affinity. The cells then move into another region and compete for antigen displayed on follicular dendritic cells. Despite the similar name, follicular dendritic cells are not the same lineage as the dendritic cells that activate naive T cells. They are stromal cells that hold antigen and immune complexes on their surfaces for B cells to test. A B cell whose mutated receptor captures antigen effectively can process it and present peptide to T follicular helper cells. The helper cell supplies survival and proliferation signals. A lower affinity B cell receives less help and dies. The cycle repeats. Mutation, competition, selection. This is affinity maturation. Across days and weeks, the average binding strength of the antibody response can improve dramatically. The body is not predicting the best molecular shape from first principles. It is generating variants and testing them inside the lymph node. The same enzyme helps B cells change antibody class through class switch recombination. The variable region can remain directed toward the same antigen while the constant region changes. The constant region determines how the antibody communicates with the rest of the immune system. Immunoglobulin M is often prominent early and activates complement efficiently. Immunoglobulin G becomes abundant in blood and tissues, supports neutralization and opsonization, and can cross the placenta through specialized transport, providing temporary protection to a fetus. Immunoglobulin A is specialized for mucosal surfaces and secretions. In its secretory form, it often appears as a dimer protected from degradation while crossing epithelium. Immunoglobulin E binds tightly to receptors on mast cells and basophils, contributing to defense against parasites and to immediate allergy. Immunoglobulin D is expressed mainly on naive B cells and has less clearly defined secreted roles. The classes are not simply stronger and weaker antibodies. They send the same specificity into different anatomical and functional systems. An antibody can protect in several ways. Neutralization prevents a virus or toxin from binding its target. Opsonization coats a particle so phagocytes can recognize the antibody's Fc region. Complement activation amplifies deposition and inflammation. Antibody-dependent cellular cytotoxicity allows NK cells and other effectors to detect antibody-coated targets. Agglutination links particles together and can make clearance easier. At mucosal surfaces, immunoglobulin A can exclude microbes without triggering the destructive inflammation that would damage a delicate barrier. Antibodies do not enter the interior of an ordinary intact cell efficiently. A virus hidden inside a cell must be addressed mainly through infected cell detection, interferons, and cytotoxic lymphocytes. The immune system divides the problem according to compartment. Extracellular target, intracellular target, barrier surface, bloodstream. Each location favors different effectors. Some germinal center B cells become plasma cells that migrate to bone marrow and secrete antibodies for years or decades. Others become memory B cells. Memory does not mean one permanent, unchanged copy of the original response. Memory populations are diverse. Some retain immunoglobulin M. Some have switched class. Some carry highly matured receptors. On re-exposure, they can respond quickly, re-enter germinal centers, or produce plasma cells. T-cell memory is also diverse. Central memory cells circulate through lymphoid organs. Effector memory cells survey blood and peripheral tissues. Tissue-resident memory cells remain in skin, lung, intestine, and other sites, positioned near the route through which the pathogen first entered. A later encounter can therefore begin with experienced cells already present. Immunological memory is often described as a record. It is more like a changed population. The body does not store an image of the pathogen in a central archive. It keeps cells whose receptors, locations, numbers, and epigenetic states were altered by the previous encounter. Memory can last for decades, but duration differs among pathogens, vaccines, cell types, and individuals. Some antibody levels decline while memory B cells remain. Some infections produce strong protection against severe disease, but weaker protection against reinfection at a mucosal surface. Some pathogens mutate their exposed proteins or interfere with memory formation. Some, such as measles, can damage existing immune memory by infecting immune cells, a phenomenon sometimes called immune amnesia. The phrase immunity is therefore not one binary state. Protection can mean preventing infection, reducing symptoms, preventing severe disease, shortening contagiousness, or responding more effectively without complete sterilizing immunity. At the splinter, new antibodies enter the inflamed tissue from blood. They bind bacterial surfaces, complement deposits more efficiently. Macrophages and neutrophils recognize the coding. T cells provide cytokines that strengthen selected antimicrobial functions. If bacteria entered host cells, cytotoxic T cells can identify infected cells displaying bacterial or viral peptides. The adaptive response has increased specificity and scale. It has also increased the risk of collateral damage. The same antibody can form immune complexes. The same T cell can attack healthy tissue if its target is shared or mimicked. The activated lymphocytes do not all become one interchangeable weapon. Before the response contracts, it is worth seeing why the dendritic cell carried information about context as well as antigen. The splinter presents mainly extracellular bacteria entering damaged tissue. A virus creates a different geometry. A virus cannot reproduce on its own. It enters a host cell and uses that cell's machinery to copy genetic material, produce proteins, and assemble new particles. Once inside, the pathogen is hidden behind a membrane carrying the body's own surface molecules. The immune system must recognize infection without destroying every nearby uninfected cell. The infected cell often detects the invasion first. Viral RNA or DNA appears in unusual forms or compartments. RIG1-like receptors, Toll-like receptors, CGASSTING pathways, and other sensors activate transcription of type 1 and type 3 interferons. The name interferon came from the observation that cells exposed to one virus released a factor that interfered with the growth of another. Interferons bind receptors on the producing cell and its neighbors. Hundreds of interferon-stimulated genes change the cellular environment. Some degrade viral RNA. Some inhibit translation. Some alter membranes or nucleotide metabolism. Some increase antigen presentation. The neighboring cells enter an antiviral state before the virus reaches them. Interferon is a warning system with a cost. It can slow normal cell growth, change metabolism, and contribute to fever, fatigue, and tissue injury. Viruses evolve proteins that block production, signaling, or antiviral effectors. The immune system strengthens one pathway. The virus finds another point to interrupt. Natural killer cells respond during the interval before a large antigen-specific T cell clone has formed. They integrate activating and inhibitory signals. Normal MHC class I molecules engage inhibitory receptors and help mark a cell as healthy self. Viral infection and malignant transformation can reduce MHC class I in an attempt to evade cytotoxic T cells. The missing inhibitory signal makes the cell more vulnerable to NK cell killing. This is missing self-recognition. The phrase is useful without being complete. NK cells also detect stress-induced ligands and antibodies attached to target cells. Cytokines such as interleukin-12, interleukin-15, and type 1 interferons alter their activation. They release perforin and granzymes, much like cytotoxic T cells. They can also release interferon gamma and influence macrophages and dendritic cells. A virus trying to hide from one lymphocyte may expose itself to another. Later, CD8 T cells expand against viral peptides and search tissues for infected cells displaying the correct peptide-MHC combination. Antibodies neutralize particles outside cells and prevent new infection. The two arms meet at different stages of the viral life cycle. Antibody blocks entry. Cytotoxic cells remove infected factories. Memory at a mucosal surface can respond rapidly, but many respiratory viruses replicate in epithelial cells before circulating antibody reaches high concentration. This is one reason a vaccine may protect strongly against severe disease while allowing some infection. The pathogen begins at the surface. Memory may become fully effective after the first rounds of replication. Viruses also alter their antigens. RNA viruses often mutate rapidly because replication enzymes can make errors and large populations create many variants. Influenza changes gradually through antigenic drift and occasionally more dramatically through reassortment of genome segments. SARS-CoV-2 variants changed spike protein regions recognized by antibodies while retaining other targets for T cells. Immune escape is rarely all or nothing. An antibody may bind less strongly. A memory B cell may still recognize a conserved region and mature further. A T cell may recognize peptides unaffected by the mutation. Protection shifts rather than vanishing in one instant. Some viruses avoid clearance by becoming latent. Herpes viruses maintain their genomes inside long-lived cells and reactivate intermittently. Epstein-Barr virus persists in B cells. Varicella-zoster virus remains in sensory ganglia after chickenpox and can later reactivate as shingles. Cytomegalovirus occupies a large part of the memory T cell repertoire in many older adults. The immune system does not always eliminate an infection. It can hold it in a long-term equilibrium. If immunity weakens, the equilibrium changes. Fungi create another set of demands. Their cells are eukaryotic like human cells, making selective drug and immune targeting harder than for bacteria. Their walls contain glucans and monons recognized by C-type lectin receptors. Neutrophils are especially important against invasive molds and yeasts. A small fungal cell can be phagocytosed. A long hypha may be too large, requiring extracellular release of reactive molecules and enzymes. Patients with prolonged neutropenia are therefore vulnerable to invasive fungal infection. T helper 17 responses support mucosal defense, and defects in related pathways can produce chronic candida infection. A pathway associated with autoimmune disease when excessive is essential against fungi when properly controlled. Helminth parasites are larger still. A worm cannot be swallowed by one phagocyte. Type 2 immunity recruits eosinophils, mast cells, immunoglobulin E, mucus, smooth muscle contraction, and tissue repair programs. The response can help expel or contain parasites. It also underlies allergy and can cause fibrosis. The machinery used against large multicellular invaders becomes harmful when directed toward pollen or food. Protozoan parasites occupy varied compartments. Malaria parasites move from liver cells into red blood cells, which lack nuclei and ordinary MHC class I presentation. The parasite changes stage, antigen, and location. Antibodies, T cells, spleen, macrophages, and inflammatory pathways each act at different times. The complexity of the life cycle makes durable vaccine protection difficult. The immune system has no single weapon called immunity. Different organisms force different compromises. An extracellular bacterium can be coated and engulfed. An intracellular virus requires altered host cells to be recognized. A fungus may require neutrophils and barrier T cells. A helminth requires containment or expulsion rather than phagocytosis. The response most effective against one class can be ineffective or damaging against another. This specialization begins with the innate signals carried by dendritic cells and continues through helper T cell differentiation, antibody class, tissue location, and memory. Pathogens in turn manipulate these decisions. Some bacteria inject proteins that block phagocytosis. Some survive inside phagosomes or escape into the cytoplasm. Some produce capsules that resist complement and engulfment. Viruses block interferon, antigen presentation, or cell death. Parasites vary surface antigens or suppress local immunity. The immune system and pathogens have evolved in response to one another for hundreds of millions of years. Many immune mechanisms that appear unnecessarily complicated are answers to earlier microbial evasions. Many microbial mechanisms are answers to earlier immunity. The relationship is not an arms race moving toward one final winner. Pathogens that kill every host too quickly may lose opportunities to spread. Hosts that damage themselves during every minor infection may reproduce less successfully. Evolution favors transmission, survival, and trade-offs rather than perfection. This helps explain why symptoms can be produced by both sides. A cough can spread a respiratory virus and clear mucus from the host. Diarrhea can expel pathogens and transmit them. Fever can inhibit microbes and stress the body. Inflammation can contain infection and injure tissue. Disease is the combined result of pathogen damage, immune response, organ reserve, and timing. The same virus causes a mild illness in one person and severe disease in another because the encounter occurs inside different immune histories and bodies. Age, genetics, previous exposure, dose, route, microbiome, medication, metabolic and cardiovascular health, chance in the first infected cells. No one factor explains every outcome. The immune system provides probability, not invulnerability. The splinter wound has required only one branch of this larger repertoire. Its extracellular bacteria can be coated, engulfed, and contained while the barrier closes. Now the enlarged response must become smaller. The same cytokines that activate macrophages can injure organs when persistent. The response must contract. When antigen and inflammatory signals fall, survival factors become scarce. Most effector lymphocytes die through apoptosis. Macrophages clear them. A smaller memory population remains. The lymph node gradually returns toward its earlier architecture. The clone that expanded so dramatically becomes rare again, but not as rare as before. The encounter has changed the body. Vaccination uses this change deliberately. It creates immune memory while avoiding, as far as possible, the damage of the disease itself. The idea of inducing protection before exposure is older than modern immunology. Long before viruses were seen, people in parts of Asia, Africa, and the Ottoman world practiced forms of variolation against smallpox. Material from a smallpox lesion was introduced through the skin or nose of a person who had not had the disease. The procedure could produce a milder infection and later protection. It also carried serious risk. The recipient could develop full smallpox, die, or spread the virus to others. In the early 18th century, Lady Mary Wortley Montagu observed inoculation in the Ottoman Empire after surviving smallpox herself. She had her son inoculated and helped introduce the practice more widely in Britain. The history involved local practitioners, enslaved Africans who described related knowledge in the Americas, physicians, families, and intense public argument. Edward Jenner's later work created a safer route. In 1796, Jenner inoculated James Phipps with material from a cowpox lesion and later exposed him to smallpox material under practices that would be ethically unacceptable today. The boy did not develop smallpox. Jenner did not discover vaccination from nothing, and the observation that cowpox could protect against smallpox circulated among rural communities before his experiment. He systematized, tested, and promoted the approach. The word vaccination came from vacca, the Latin word for cow. More than a century and a half later, coordinated vaccination and surveillance eliminated smallpox from natural transmission worldwide. The World Health Organization declared eradication in 1980. The immune system had become part of public infrastructure. A vaccine must provide antigen and enough contextual information to produce useful memory. Live attenuated vaccines use a weakened form of a pathogen capable of limited replication. They often stimulate broad and durable responses because antigen appears in several cellular compartments and persists long enough to imitate aspects of infection. The same feature can make them unsuitable for some people with severe immune suppression. Inactivated vaccines contain killed organisms and cannot replicate. but may need repeated doses or adjuvants. Protein subunit vaccines deliver selected antigens rather than the whole pathogen. Conjugate vaccines attach bacterial polysaccharides to carrier proteins, allowing young children to recruit T-cell help and form stronger memory against capsules that otherwise stimulate a limited response. Viral vector vaccines use a modified virus to deliver genetic instructions. Messenger RNA vaccines deliver messenger RNA encoding an antigen inside lipid nanoparticles. Cells translate the messenger RNA temporarily, make the antigen, and present it to the immune system. The messenger RNA does not need to enter the nucleus and is broken down through ordinary cellular processes. The platform became widely visible during the COVID-19 pandemic, but it was built on decades of research into RNA chemistry, delivery, innate sensing, and antigen design. In 2024, the first messenger RNA vaccine approved in the United States for a pathogen other than SARS-CoV-2 was authorized against respiratory syncytial virus in older adults. Its antigen is the RSV fusion protein stabilized in the prefusion form. That detail reveals another advance. A vaccine antigen is not always chosen as a natural protein in any shape. Structural biology can identify the form present before a virus fuses with a cell, preserve that form, and focus antibodies on vulnerable surfaces. The immune system performs the selection. Molecular engineering decides what it is allowed to see. Adjuvants provide another form of context. Aluminum salts have been used for decades. Newer adjuvants contain emulsions, lipids, or define molecules that activate particular innate pathways. They can increase antibody levels, improve durability, reduce the amount of antigen needed, or shift the kind of T-cell help produced. The old idea that an adjuvant simply irritates tissue is incomplete. An adjuvant helps determine which innate cells activate, which cytokines appear, how antigen is retained or transported, and what kind of adaptive memory follows. Vaccination is therefore controlled communication between innate and adaptive immunity. A sore arm, fatigue, or mild fever after vaccination can reflect this temporary inflammatory program. These symptoms are not required for protection and do not measure protection precisely. A person can form strong memory with few symptoms, while intense symptoms do not guarantee a stronger response. The immune system varies with age, genetics, previous exposure, medication, sleep, and health. Booster doses serve several purposes. They can restore antibody levels that declined. They can expand memory cells. They can drive further germinal center selection and broaden recognition. An updated antigen can redirect memory toward a changed pathogen. But repeated exposure also interacts with the existing response. The immune system does not start from zero each time. Earlier antibodies and memory B cells shape which clones compete. This can accelerate protection and can sometimes bias the response toward familiar regions of a pathogen. The concept is called immune imprinting. It is not automatically harmful. It describes how history constrains the next response. Adaptive memory is no longer considered the only form of altered future response. Innate cells and their bone marrow progenitors can also interchange functional states after certain infections, vaccines, or microbial products. Epigenetic modifications and metabolic rewiring can make later responses stronger or different even without antigen-specific B or T cell receptors. This phenomenon is called trained immunity. It does not create the same precise memory as an antibody clone. Its effects can be broader and shorter-lived, and they can be beneficial or harmful depending on context. Training may improve resistance to unrelated infections. Persistent maladaptive training may contribute to chronic inflammation. The boundary between innate and adaptive immunity is therefore less absolute than the old diagram suggests. The systems remain distinct in receptor generation and specificity, but both can be shaped by experience. Vaccines also reveal a social property of immunity. One person's protection can reduce transmission to others. When enough people are immune to a pathogen with no important non-human reservoir, chains of infection become difficult to sustain. This population effect protects people who cannot be vaccinated or who respond poorly. Its threshold depends on contagiousness, vaccine effectiveness against infection, behavior, and the distribution of immunity. It is not one fixed percentage for all diseases. Immunity exists in cells. Public health emerges from how those cells are distributed across communities. The success of a vaccine can make the disease it prevents disappear from ordinary memory. That success creates a paradox. As the disease becomes less visible, rare adverse effects or fear of the intervention may feel more immediate than the infection itself. The immune system remembers even when society forgets. The next problem is the opposite of weak memory. What happens when immune recognition is directed toward harmless material or toward the body itself? Allergy begins when a harmless substance is given the meaning of immediate danger. The substance may be a pollen protein, a food protein, dust mite material, an animal protein, a medicine, or insect venom. The molecule is not dangerous in the same way for everyone. The danger lies in the response it triggers in a sensitized person. During sensitization, antigen is presented in a context that favors a type 2 immune response. T helper 2 cells produce cytokines that encourage B cells to switch toward immunoglobulin E. Immunoglobulin E circulates at low concentration compared with immunoglobulin G, but it binds with very high affinity to Fc receptors on mast cells and basophils. The antibodies can remain attached to those cells for long periods. The tissue is now prepared. On later exposure, one allergen molecule can bind and cross-link several adjacent immunoglobulin E molecules. The mast cell releases histamine and other mediators. Vessels dilate and become more permeable. Smooth muscle can contract. Mucus increases. Nerve endings produce itch and sneezing. In the nose, the result may be allergic rhinitis. In the skin, hives. In the airways, bronchoconstriction and asthma symptoms. A later phase recruits eosinophils, T cells, and additional inflammation, prolonging tissue damage after the immediate histamine release is faded. Anaphylaxis is the systemic version. Mediators released across many tissues can dilate vessels, lower blood pressure, cause fluid leakage, constrict airways, and produce gastrointestinal and skin symptoms within minutes. Epinephrine is the first-line emergency treatment because it reverses several of these changes at once. It constricts vessels, supports blood pressure, relaxes airway smooth muscle, and reduces further mediator release. Antihistamines can help some symptoms, but cannot replace epinephrine during anaphylaxis. Why one person develops immunoglobulin E to peanut while another develops ordinary tolerance remains a question with many interacting answers. Genetic variants alter skin barrier proteins, cytokine pathways, and receptor signaling. Early eczema can allow food proteins to enter through inflamed skin before they are encountered through the gut. Microbial exposure, diet, pollutants, infections, and timing influence immune development. The old hygiene hypothesis suggested that reduced childhood infection in industrialized societies allowed allergic disease to rise. The modern view is more precise and less romantic about infection. Children do not need dangerous diseases in order to train immunity properly. They may benefit from exposure to diverse, largely harmless microorganisms associated with people, animals, soil, food, and stable microbial communities. Barrier integrity matters. Antibiotics and diet can alter the microbiome. Urban environments can reduce some exposures while increasing pollution. Allergy probably reflects several changing systems rather than one missing childhood infection. Tolerance can sometimes be rebuilt. Allergen immunotherapy exposes a person to controlled amounts through injections or under the tongue over time. The response can shift away from immunoglobulin E-dominated reactivity toward regulatory pathways and blocking antibodies. Oral immunotherapy for food allergy can raise the amount needed to trigger a reaction in selected patients, reducing risk from accidental exposure. It does not always create permanent freedom from allergy, and treatment itself can cause reactions. The immune system can be retrained, but not without supervision or risk. Autoimmune disease turns recognition towards self-tissue. The phrase immune system attacks the body is true at a distance and incomplete in detail. Different diseases involve different targets, cells, antibodies, organs, and initiating conditions. In type 1 diabetes, immune cells destroy insulin-producing beta cells in pancreatic islets. In multiple sclerosis, inflammatory responses damage myelin and other structures in the central nervous system. In rheumatoid arthritis, immune complexes, antibodies, T cells, macrophages, fibroblasts, and bone remodeling cells produce persistent inflammation in joints. In systemic lupus erythematosus, antibodies against nuclear material form complexes that deposit and activate complement in several organs. The target may be one molecule. The disease is an ecosystem. Genetics creates susceptibility without usually determining certainty. HLA variants influence which peptides are presented. Variants in cytokines, complement, clearance pathways, and regulatory cells alter thresholds. Infection, smoking, ultraviolet light, hormones, medication, tissue damage, or microbiome changes can contribute. A pathogen may carry a peptide resembling a self-peptide, creating molecular mimicry. Inflammation can expose normally hidden antigens. Tissue destruction can release more targets and broaden the response through epitope spreading. A feedback loop develops. Damage creates antigen. Antigen creates more response. Loss of tolerance does not mean every checkpoint failed at once. A self-reactive cell may escape thymic or bone marrow deletion, remain quiet for years, then receive abnormal co-stimulation in an inflamed tissue. A regulatory T cell population may be insufficient locally. A B cell may receive help because a microbial and cell structure are linked. Clearance of dying cells may fail, allowing nuclear material to remain visible. 1 week's safeguard may be compensated by another until several conditions coincide. This layered protection explains why autoimmunity can appear after years of ordinary health. Treatment often works by reducing one part of the network. Corticosteroids broadly change gene expression and suppress inflammation. Methotrexate alters proliferating and signaling pathways. Antibodies can block tumor necrosis factor, interleukin-6 receptors, B-cell proteins, co-stimulation, or cell trafficking. Small molecules can inhibit Janus kinases and other intracellular pathways. These treatments can restore function and prevent damage. They also reveal the trade-off. A pathway driving autoimmunity may also protect against infection or cancer. The aim is rarely to turn immunity off. It is to lower the harmful response enough while preserving useful defense. Newer therapies attempt deeper resets. CD19-directed CAR-T cells were developed to remove malignant B cells. Beginning with small case series, researchers used them in severe autoimmune diseases such as lupus, systemic sclerosis, and inflammatory myositis. The engineered T cells removed B lineage cells broadly. As the B cell population later returned, some patients remained in remission without their previous immunosuppressive treatment. The results suggested that destroying an autoreactive B cell system might allow a more tolerant repertoire to regrow. The evidence is promising and still investigational. Early case series cannot establish long-term safety, durability, or which patients benefit most. CAR-T therapy can cause cytokine release syndrome infection, neurologic toxicity, and prolonged immune suppression. A dramatic remission is not yet a routine autoimmune treatment. The experiment, nevertheless, turns tolerance into a new question. Can an immune system be rebooted? The answer may differ by disease. In one condition, autoreactive antibodies dominate. In another, tissue-resident T cells or innate inflammation continue after B cells disappear. Resetting one compartment may be enough, temporary, or irrelevant. The immune system has no single reset button because it has no single control center. Its decisions are distributed across organs and cell populations. The gut contains the largest and most continuous version of that negotiation. The intestine contains food, digestive enzymes, bacterial metabolites, viruses, fungi, and trillions of microorganisms separated from internal tissue by a thin epithelial surface. The immune system cannot solve this environment by sterilizing it. A sterile intestine would not remain sterile for long, and many resident organisms contribute to digestion, vitamin production, barrier function, and immune development. The goal is controlled coexistence. The microbial community varies along the digestive tract. Acid, oxygen, bile, transit time, mucus, diet, and anatomy create different habitats. The colon contains the densest populations. Most organisms remain within the lumen and outer mucus. Epithelial cells and immune cells manage the boundary. Goblet cells secrete mucus. Paneth cells in the small intestine release antimicrobial peptides. Specialized M cells transport samples from the lumen to immune structures such as Peyer's patches. Dendritic cells and macrophages sample material beneath the epithelium. Innate lymphoid cells respond rapidly to cytokines and help maintain barrier tissues. Their organization mirrors some helper T cell programs without using rearranged antigen-specific receptors. Type 3 innate lymphoid cells can produce interleukin-22, supporting epithelial repair and antimicrobial defenses. Too little signaling weakens the barrier. Too much contributes to inflammation. Secretory immunoglobulin A provides a gentle form of containment. Plasma cells beneath the epithelium release dimeric immunoglobulin A. An epithelial receptor carries it across the cell and releases it into mucus with a protective component attached. Immunoglobulin A binds microbes and toxins, limits attachment, and can influence which organisms occupy the niche. It often performs these functions without strongly activating complement or recruiting destructive cells. The antibody is specific. The response is restrained. Regulatory T cells are abundant in the gut. Some are induced by food antigens or microbial products. Certain bacterial metabolites, including short-chain fatty acids produced when microbes ferment dietary fiber, can influence regulatory cell development, epithelial metabolism, and inflammation. These findings have encouraged claims that one food or one bacterial species can control immunity. The real system is less direct. Microbial metabolites depend on community composition, substrate, host genetics, transit, and tissue state. An association between one organism and one disease does not prove that adding or removing that organism will cure the disease. The microbiome is an ecosystem, not a list of supplements. Antibiotics reveal its resilience and vulnerability. A course of antibiotics can save a life by treating bacterial infection. It can also reduce microbial diversity, remove competitors, and allow resistant organisms or Clostridioides difficile to expand. Most communities recover partly. Some changes persist. The effect depends on drug, duration, age, diet, and previous exposures. The lesson is not that antibiotics are bad. It is that killing bacteria changes an ecological system as well as a pathogen. Inflammatory bowel disease emerges when barrier, microbiome, innate sensing, and adaptive immunity enter a self-sustaining inflammatory state. Crohn's disease can affect different parts of the digestive tract and penetrate deeply through the bowel wall. Ulcerative colitis primarily affects the colon's mucosa. Neither disease is simply an immune reaction to one invading organism. Genetic susceptibility alters microbial sensing, autophagy, epithelial defense, and cytokine pathways. The microbiome changes in the inflamed environment. Inflammation itself favors different organisms. Cause and consequence become entangled. Therapies target tumor necrosis factor, integrins controlling cell migration, interleukin pathways, Janus kinases, and other mechanisms. Diet and microbial treatments are being studied, but no universal microbiome correction exists. The gut also communicates with the nervous system. The phrase gut-brain axis covers several routes. Microbial metabolites enter circulation. Immune cytokines affect nerves and brain barriers. The vagus nerve carries signals in both directions. Enteroendocrine cells release hormones. Stress hormones alter motility, permeability, and immune responses. These connections are real and often exaggerated into claims that one microbial change directly determines mood or personality. Human mental health emerges from brain circuits, experience, genetics, hormones, immunity, social conditions, and many other factors. The gut participates. It does not act alone. The brain itself is not immunologically empty. For many years, it was described as immune privileged because ordinary immune traffic is restricted, lymphatic drainage differs, and inflammation can be especially damaging inside a rigid skull. The phrase was sometimes misunderstood to mean that the brain lacks immunity. Microglia live throughout neural tissue. They arise early in development and maintain themselves locally. They remove debris, respond to infection and injury, sculpt synapses during development, and alter their states in neurological disease. Border-associated macrophages occupy meninges, vessels, and other interfaces. T cells patrol cerebrospinal fluid and meningeal compartments. Lymphatic vessels in the meninges connect central nervous system drainage with cervical lymph nodes. The brain is protected through specialized access and regulation, not isolation. Neuroinflammation can protect against infection and help repair damage. Persistent or misdirected activation can contribute to multiple sclerosis, neurodegeneration, pain, and cognitive symptoms. Microglia near an amyloid plaque, a damaged axon, or a virus do not have one universally good or bad state. They can remove material, release toxic mediators, support neurons, or become dysfunctional depending on disease stage and local signals. The old division between nervous and immune systems continues to dissolve. Sleep is one place where their communication becomes visible. Immune signals can increase sleepiness during infection. Sleep and circadian rhythms in turn alter leukocyte trafficking, cytokine production, antibody responses, and inflammation. One poor night does not cause the immune system to collapse. Chronic sleep disruption can shift several immune measures and is associated with weaker vaccine responses and greater inflammatory activity in some studies. The relationship runs in both directions. Inflammation changes sleep. Sleep changes inflammation. The immune system working tonight is therefore not performing the same tasks at every hour. Cells enter and leave blood according to circadian signals. Hormones change receptor expression. Lymphocytes redistribute between circulation and tissues. Night is not a shutdown. It is another phase of coordination. The immune system also watches for threats arising without infection. Every cell division carries a small chance of mutation. Some altered cells acquire the ability to grow despite signals that should restrain them. Cancer begins within self, which makes immune recognition especially difficult. Cancer cells are descended from the body's own cells. They carry ordinary HLA molecules, ordinary membrane lipids, and many ordinary proteins. The difference can be subtle. A mutation creates a new peptide. A developmental protein appears at the wrong age. A growth receptor becomes unusually abundant. A virus contributes foreign proteins. Stress pathways place activating ligands on the surface. The immune system can recognize some of these changes. The idea is called cancer immunosurveillance. Cytotoxic T cells can detect tumor peptides on MHC class I. Natural killer cells can detect cells that lose MHC or express stress signals. Macrophages and antibodies can contribute. Immune deficiency increases the risk of selected cancers, especially cancers associated with viruses. Transplanted organs require immune suppression, and recipients have higher risk of lymphomas and skin cancers. These observations show that immunity prevents some malignant growth. They do not mean every small tumor is automatically eliminated. Cancer evolves under selection. A cell recognized strongly may be killed. A neighboring cell that presents less antigen, loses MHC, recruits suppressive cells, or resists apoptosis may survive and expand. The immune response shapes the tumor population. This process is called immunoediting. It can be described in three broad phases. Elimination, when immunity removes transformed cells. Equilibrium, when surviving cells are contained while continuing to change. Escape, when variants grow despite the response. A visible cancer has often already passed through selection imposed by the immune system. Tumors create local environments that suppress attack. They consume glucose and oxygen. They produce adenosine, lactate, transforming growth factor beta, and other mediators. They recruit regulatory T cells, suppressive myeloid cells, and macrophages that support blood vessels and tissue remodeling. They exclude T cells physically or chemically. They can express ligands that activate inhibitory receptors on lymphocytes. The immune system contains these inhibitory receptors for good reasons. A T cell must stop after infection. Repeated stimulation without restraint can injure tissue. Checkpoints reduce activation, maintain tolerance, and limit chronic inflammation. Cancer uses the brakes that evolved to protect the body. James Allison studied one such brake, CTLA4. Many researchers viewed CTLA4 mainly as a molecule involved in T cell regulation. Allison asked whether blocking it could release an anti-tumor response already present. In mouse experiments, antibodies against CTLA4 allowed T cells to reject tumors. The approach was unusual because the antibody did not target the cancer cell directly. It targeted inhibition. The treatment asked the immune system to do what it had previously failed to do. Tasuka Honjo discovered another inhibitory receptor, PD-1. Its ligands, including PD-L1, can be expressed by tumor and immune cells. Blocking the pathway restored T cell activity in selected cancers. CTLA4 and PD-1 do not operate identically. CTLA4 strongly influences early activation and competition for co-stimulation. PD-1 restrains chronically stimulated cells in tissues and tumors. Drugs blocking these pathways produced long-lasting remissions in some patients with melanoma, lung cancer, kidney cancer, lymphoma, and other malignancies. In 2018, Allison and Honjo received the Nobel Prize for Cancer Therapy by inhibition of negative immune regulation. The phrase releasing the brakes became common. It should not imply that more immunity is always better. Checkpoint inhibitors can cause immune-related adverse events resembling autoimmune disease. T cells can inflame the colon, liver, lungs, skin, endocrine glands, heart, nerves, and other organs. Some effects resolve with corticosteroids or other immune suppression. Others cause permanent hormone deficiencies or severe injury. The same mechanism that allows tumor killing can remove tolerance. Response also varies widely. Some tumors contain many mutations and pre-existing T cells, and respond well. Others lack recognizable antigens, exclude lymphocytes, or use alternative suppressive pathways. Biomarkers such as PD-L1 expression, mismatch repair deficiency, and tumor mutational burden help in selected contexts but do not predict every patient accurately. Immunotherapy is not one universal cure. It is a new principle with uneven effects. CAR T-cell therapy takes a different approach. T-cells are collected from a patient and genetically modified to express a chimeric antigen receptor. The receptor combines an antibody-like binding domain with intracellular T-cell signaling domains. It can recognize a surface molecule without requiring peptide presentation by MHC. The cells are expanded and returned after preparative treatment. For B-cell leukemias and lymphomas, CD19 became a powerful target because malignant and normal B lineage cells express it. CAR T cells can expand in the body and eliminate enormous numbers of cancer cells. Some patients with otherwise refractory disease achieve durable remission. The treatment can also produce cytokine release syndrome. Activated cells release interferon and other signals, stimulating monocytes and widespread inflammation. Fever, low blood pressure, low oxygen, and organ dysfunction can follow. Blocking interleukin-6 signaling and providing supportive care has made the syndrome more manageable, but severe cases remain dangerous. Neurologic toxicity can produce confusion, seizures, language difficulty, or cerebral edema. Eliminating normal B cells increases infection risk and can require antibody replacement. Cancer can escape by losing or changing the target antigen. Solid tumors are harder because their antigens are less exclusive, their structure restricts access, and their environment suppresses T cells. Researchers are designing dual target receptors, controllable switches, armored cells producing cytokines, and CAR cells based on other immune lineages. Tumor-infiltrating lymphocyte therapy uses cells already present inside a tumor. A surgeon removes tumor tissue. T cells are isolated and expanded in very large numbers outside the body. After lymphodepleting treatment, the cells are returned with growth support. In 2024, the United States approved lifileucel for selected adults with advanced melanoma previously treated with other therapies. It was the first approved cellular therapy using a patient's own tumor-derived T cells. The treatment preserves a diverse population of receptors rather than engineering one shared receptor. The tumor has already selected the cells by attracting them. The laboratory gives them number and another opportunity. Cancer vaccines attempt to teach immunity which tumor features to recognize. Some prevent cancer by preventing infection, as vaccines against human papillomavirus and hepatitis B do. Therapeutic vaccines target an existing tumor. Personalized approaches sequence a tumor, identify mutations likely to produce presented peptides, and create a vaccine containing selected neoantigens, sometimes using messenger RNA. The aim is not merely to produce antibody. It is to expand T-cell clones against mutations unique to that tumor. Trials have produced encouraging results in selected cancers, often combined with checkpoint blockade. The field remains experimental for many applications. Cancer immunology reveals the system's deepest ambiguity. A strong response can save a life. The same response can damage healthy organs. A tumor is self enough to hide and altered enough to be targeted. Treatment succeeds by changing thresholds, cell numbers, location, or inhibition. The immune system is not replaced. It is persuaded, engineered, or released. Another medical field confronts the same power from the opposite direction. Transplantation asks the immune system to tolerate healthy tissue that is genuinely foreign. An organ transplant carries cells displaying the donor's HLA molecules. The recipient's T-cell repertoire was selected to interact with the recipient's own HLA. donor HLA can be recognized with unusual strength and frequency. Some T cells bind donor molecules directly, treating the foreign MHC peptide surface as though it were a familiar MHC carrying a dangerous peptide. Other recipient antigen-presenting cells process donor proteins and present fragments indirectly. Antibodies can bind blood group or HLA antigens on donor vessels. The transplanted organ may function perfectly and still appear immunologically alarming. Hyperacute rejection can occur within minutes or hours when pre-existing antibodies bind the graft's vessels, activate complement, and cause thrombosis. Modern blood group matching and antibody screening make this uncommon. Acute rejection develops over days to months through T cells and newly produced antibodies. Chronic rejection unfolds across years through vascular narrowing, fibrosis, repeated injury, and several immune and non-immune factors. Matching donor and recipient HLA reduces risk. Immunosuppressive medicines restrain T cell activation, proliferation, cytokine signaling, or antibody production. Calcineurin inhibitors block a pathway needed for interleukin-2 transcription. Antimetabolites reduce lymphocyte proliferation. Corticosteroids suppress broad inflammatory programs. Antibodies can remove lymphocytes or block co-stimulation. The medicines make transplantation possible. They also increase infection, cancer, metabolic, kidney, and cardiovascular risks. The recipient must live between rejection and excessive immune suppression. A transplant can also carry its own immune cells. In bone marrow or hematopoietic stem cell transplantation, donor T cells may attack the recipient's tissues, producing graft versus host disease. The direction has reversed. The graft rejects the host. The same donor cells can also attack residual leukemia, creating a valuable graft versus leukemia effect. Doctors attempt to preserve one response while limiting the other. Again, the useful and harmful functions share mechanisms. The long-term goal is transplant tolerance, accepting the graft without continuous generalized suppression. Researchers use mixed chimerism, regulatory cells, co-stimulation blockade, and engineered tissues to try to teach or rebuild tolerance. Rare patients have achieved operational tolerance under selected protocols. No universal method exists. Transplantation makes immune identity visible because two healthy bodies cannot exchange most organs without negotiation. Immunodeficiency reveals what happens when parts of the system are absent. Primary immunodeficiencies, increasingly called inborn errors of immunity, arise from inherited variants affecting immune development or function. The clinical pattern can reveal the missing mechanism. A person unable to produce antibodies develops repeated infections with encapsulated extracellular bacteria, especially after maternal antibodies decline. A severe T-cell defect can cause vulnerability to viruses, fungi, protozoa, and live vaccines. A phagocyte unable to generate a respiratory burst develops characteristic bacterial and fungal infections and granulomas. A terminal complement deficiency creates recurrent Neisseria infection. Some variants produce not only infection but autoimmunity, allergy, or uncontrolled inflammation. A broken immune gene does not always make immunity simply weaker. It can remove one restraint and make part of the system stronger in the wrong direction. Severe combined immunodeficiency prevents effective adaptive immunity from developing. Infants can appear healthy initially because maternal immunoglobulin G crossed the placenta, but then develop persistent infections, diarrhea, poor growth, and unusual organisms. Newborn screening measures DNA circles produced during T-cell receptor rearrangement, allowing some cases to be detected before infection begins. Treatment can include hematopoietic stem cell transplantation, enzyme replacement for selected forms, or gene therapy. Gene therapy returns to the origin of the immune system. Stem cells are collected. A functional gene is inserted or repaired. The corrected cells repopulate blood lineages. Early gene therapy trials for one form of severe combined immunodeficiency restored immunity, but caused leukemia in some children when the viral vector activated growth genes near its insertion site. The tragedy changed vector design and monitoring. Later approaches became safer, though no genetic intervention is without uncertainty. The immune system can be repaired at the level of one gene because all its circulating cells descend from stem and progenitor cells. Acquired immunodeficiency can arise through infection, malnutrition, cancer, medication, or aging. Human immunodeficiency virus targets cells expressing CD4 and suitable co-receptors, especially CD4 T cells. The virus integrates its genetic material into host cell DNA and establishes reservoirs that persist despite treatment. Without therapy, progressive loss and dysfunction of CD4 T cells disrupts coordination across the immune system. Macrophage activation weakens. B-cell responses become abnormal. Cytotoxic responses lose support. Organisms ordinarily controlled by cellular immunity cause opportunistic disease. HIV demonstrated that one cell population could hold the architecture together. Antiretroviral therapy blocks several stages of viral replication and allows immune recovery. A combination of drugs is used because the virus mutates rapidly and can escape one inhibitor. Effective treatment can keep the amount of virus in blood below routine detection. When that suppression is maintained, HIV is not sexually transmitted, a principle summarized as undetectable equals untransmittable. The treatment does not ordinarily remove the latent reservoir. Stopping therapy usually allows virus to return. Rare cures after stem cell transplantation from donors carrying protective CC are 5 variants showed that eradication is biologically possible, but the dangerous procedure was performed to treat life-threatening cancer and is not a practical general cure. Research explores broadly neutralizing antibodies, therapeutic vaccines, latency reversal, immune engineering, and gene editing. HIV also transformed immunology by forcing precise measurement of T cells, viral dynamics, mucosal immunity, and chronic activation. An immune response can fail because it is absent. It can also fail because it never stops responding. Chronic infection keeps antigens present and changes T cells over time. Repeatedly stimulated T cells enter states described as exhaustion. They express inhibitory receptors, alter metabolism and gene regulation, and lose some effector functions while retaining others. Exhaustion limits tissue damage during persistent infection and prevents full pathogen clearance. Cancer uses related programs. Checkpoint blockade can revive selected exhausted or suppressed T cells, but not return every cell to a youthful original state. The immune system records chronicity in cell identity. Time is one of its signals. The same is true across the human lifespan. HIV also changed how medicine learned to see the immune system. A person can carry an enormous amount of immunological activity without any structure being visible from the outside. The lymph node is hidden. Most tissue-resident cells never enter an ordinary blood sample. Cytokines rise and fall. Clones expand and contract. During the HIV epidemic, one measurement became especially important: the number of CD4 T cells in blood. The count could not describe the entire immune system. It could show that a central coordinating population was being lost and help estimate vulnerability to opportunistic infection. Producing that count relied on flow cytometry. Cells are suspended in fluid and pass through lasers one at a time. Fluorescent antibodies bind selected proteins on their surfaces or inside them. The instrument records scattered light and fluorescence, allowing a sample to be divided into populations. A cell expressing CD3 and CD4 can be distinguished from a B cell, a natural killer cell, a monocyte, or an abnormal leukemia cell. Several markers can be combined. The result is not a photograph of immunity. It is a carefully chosen coordinate system. The researcher decides which proteins to label and which gates define a population. Poor controls, damaged cells, overlapping signals, or biased gates can create a convincing pattern that is not biological. The instrument extends observation and also requires judgment. A complete blood count offers a broader first view. It reports numbers of neutrophils, lymphocytes, monocytes, eosinophils, basophils, red cells, and platelets. A high neutrophil count can accompany bacterial infection, inflammation, corticosteroid treatment, physical stress, smoking, or release of cells previously resting near vessel walls. A low count can result from marrow failure, medication, autoimmune destruction, inherited disease, or overwhelming infection. One number rarely identifies one mechanism. Serology asks whether antibodies bind a particular antigen. Depending on timing and antibody class, a positive result can reflect vaccination, old infection, recent infection, or cross-reactivity. A negative result early in disease can occur before antibodies have formed. Polymerase chain reaction asks a different question. It amplifies selected genetic material from a pathogen and can become positive before the adaptive response is measurable. Culture asks whether a living organism can grow under laboratory conditions. Each test observes a different stage of the relationship between pathogen and host. A swollen lymph node is similarly ambiguous. It may contain dividing lymphocytes responding to a throat infection. It may enlarge during autoimmune disease. It may contain malignant cells. The same outward sign can arise from very different internal stories. Modern technologies try to recover more of that hidden detail. Single-cell RNA sequencing measures which genes are active in individual cells rather than averaging an entire tissue. Receptor sequencing reconstructs T and B cell clones. Spatial methods preserve where cells and gene programs were located inside a tumor, intestine, lung, or lymph node. Protein measurements, chromatin accessibility, metabolites, and receptor sequences can be combined. The familiar labels then divide into many states. A macrophage is not one program. A regulatory T cell in the intestine is not identical to one inside a tumor. An exhausted T cell can retain some functions and lose others. More detail does not automatically produce more understanding. A dataset containing millions of measurements can reveal a population associated with recovery without showing whether the cells caused recovery, appeared because recovery had begun, or responded to a third process. Machine learning can classify patterns that no person could inspect manually. It can also inherit biased sampling and hide the biological reason for its classification. The final test remains intervention. Block the pathway, remove the population, alter the gene, then ask whether the predicted change occurs and whether it improves human health safely. Animal models remain essential because tissues can be sampled and mechanisms interrupted in ways that would be unethical in people. Mice do not reproduce every feature of human lifespan, microbiome, infection history, or disease. Human organoids, tissue cultures, challenge studies under strict safeguards, longitudinal sampling, and clinical trials each recover another part of the system. No model is the body in full. There is also no single immune system that serves as the universal normal. HLA genes vary. Age, previous infections, hormones, pregnancy, medication, nutrition, microbiome, sleep, environment, and inherited variants alter thresholds. These differences are not merely noise around one ideal design. They help a population survive varied pathogens and create different risks. A receptor variant can improve defense in one context and increase inflammatory disease in another. This is why the phrase "boost the immune system" is biologically vague. A person with autoimmunity does not need every response increased. A transplant recipient needs selected restraint. A person receiving a vaccine needs expansion of particular memory, not generalized inflammation. Medicine works by changing defined cells or signals inside a network. One of the clearest examples of an immune system changing its rules without becoming defenseless is pregnancy. Pregnancy presents a form of immune coexistence more intimate than transplantation. The fetus carries genetic material from both parents. Cells of the placenta express paternal antigens that are not identical to the mother's tissues. Yet a healthy pregnancy is not maintained by switching immunity off. The uterus must still defend against infection, repair tissue, remodel blood vessels, and later participate in labor. Tolerance is local, active, and changing across time. The placenta forms the interface. Some fetal trophoblast cells invade the uterine lining and help transform maternal spiral arteries into high-flow vessels capable of supporting growth. The trophoblast does not display the ordinary set of highly variable HLA class I and class II molecules found on most tissues. It expresses a specialized pattern, including HLA-C, HLA-E, and HLA-G, that interacts with maternal immune cells without provoking the same response as an ordinary foreign graft. Uterine natural killer cells are abundant early in pregnancy. Their name suggests that they exist mainly to kill. At this interface, many support vascular remodeling and placental development through cytokines and growth factors. Macrophages, regulatory T cells, dendritic cells, and stromal cells contribute to a controlled environment. The maternal immune system does not ignore the fetus. It forms a relationship with it. Inflammation also has scheduled roles. Implantation requires tissue remodeling and inflammatory signals. Mid-pregnancy is often relatively regulatory. Labor involves another inflammatory shift that helps contract the uterus and remodel the cervix. Too much or mistimed inflammation can contribute to pregnancy complications. Too little may impair implantation, defense, or labor. The immune state must change as the biological task changes. Pregnancy also transfers protection. Maternal immunoglobulin G crosses the placenta through the neonatal Fc receptor, especially later in gestation. After birth, antibodies in milk, particularly secretory immunoglobulin A, help protect mucosal surfaces while the infant immune system develops. The antibodies are temporary. They provide borrowed experience until the child begins building its own. Newborn immunity is not simply an immature adult system. It is adapted to transition from a largely protected fetal environment into a world filled with microorganisms. Responses are often biased away from excessive inflammation that could damage developing tissues. Maternal antibodies can protect and can sometimes reduce the infant's response to selected vaccine antigens by binding them before the infant immune system responds fully. Vaccine schedules are designed around age, risk, antibody decline, and the developing capacity for memory. The immune system has a developmental history before it has an infection history. All circulating immune cells ultimately arise from hematopoietic stem cells. These rare cells reside mainly in bone marrow niches and balance self-renewal with production of progenitors. One broad branch gives rise to lymphoid cells, including B cells, T cell precursors, and natural killer cells. Another gives rise to granulocytes, monocytes, red blood cells, and platelets, although real developmental paths form branching continua rather than one rigid tree. The marrow produces enormous numbers of cells each day. Red blood cells dominate total output. Neutrophil production can rise rapidly during infection through emergency granulopoiesis. Cytokines such as granulocyte colony stimulating factor increase proliferation, maturation, and release. The bloodstream count is therefore a snapshot of production, storage, migration, and death. Bone marrow is not an isolated factory. Sympathetic nerves and circadian signals influence stem cell release and leukocyte trafficking. Macrophages and stromal cells maintain niches through molecules such as CXCL12. Inflammation can push stem cells toward rapid myeloid production. Repeated stimulation can alter long-term output. The innate system can be trained partly by changing progenitors in marrow, so that descendants respond differently even after the original stimulus has disappeared. Experience reaches backward into production. Age changes every level of this system. The thymus begins shrinking after childhood, reducing the output of new naive T cells. Existing T cells maintain numbers through peripheral division. Memory populations accumulate. The repertoire becomes shaped increasingly by past infections, especially persistent viruses such as cytomegalovirus. Older adults can retain strong memory against familiar antigens while responding less effectively to a completely new one. B-cell generation and germinal center quality can decline. Antibodies may be produced in lower amounts or with reduced affinity after some vaccinations. Dendritic cell signaling changes. Neutrophils may migrate or kill less effectively in some contexts. Barrier repair slows. None of these changes occurs identically in every person. Biological age, infection history, nutrition, genetics, medication, and chronic disease create wide variation. At the same time, chronic low-grade inflammatory signals tend to rise. The state is called inflammaging. Senescent cells release cytokines and proteases. Damaged mitochondria and nuclear material activate innate sensors. Visceral fat produces inflammatory mediators. Barrier and microbiome changes increase exposure to microbial products. Persistent infections continue stimulating immune cells. The result is a paradox. The aging immune system can be less effective against infection and vaccination while producing more background inflammation. Weak defense and excessive activation coexist. This contributes to atherosclerosis, frailty, neurodegeneration, cancer, and metabolic disease, although immunity is one part of each condition rather than the only cause. Vaccines for older adults can compensate through higher antigen doses, adjuvants, repeated dosing, or antigens designed for the relevant pathogen. The aim is not to make an old immune system identical to a young one. It is to create sufficient protection within the system that exists. The messenger RNA RSV vaccine and other recent RSV vaccines reflect this approach to a pathogen especially dangerous at the beginning and later portions of life. Research into thymic regeneration, senescent cell removal, metabolic reprogramming, and trained immunity asks whether immune aging can be altered. No intervention has been shown to restore the entire human immune system to a youthful state safely. Suppressing inflammation too broadly can impair defense. Stimulating immunity too broadly can worsen autoimmunity or tissue damage. The same balance returns. Enough response. In the correct place. For the correct duration. Sleep participates modestly but meaningfully in that balance. During ordinary sleep, hormones and autonomic signals shift. Some leukocytes leave blood and enter lymphoid tissues. Certain cytokine patterns change. Studies have found that sleep around vaccination can influence later antibody and cellular responses, while chronic sleep restriction is associated with altered inflammation and greater susceptibility to some infections. The effects are not one simple equation in which every lost hour removes a fixed percentage of immunity. Immune function is resilient. One difficult night does not erase memory cells or antibodies. Repeated disruption changes physiology over time. The calm body at night is not an inactive body. Bone marrow continues releasing cells. Macrophages continue clearing debris. Lymphocytes continue circulating. Tissue-resident cells remain in place. Antibodies remain in fluid and mucus. Circadian timing changes which processes are emphasized. Some of the immune work associated with sleep is maintenance rather than combat. The initial splinter wound has now entered that quieter stage. The bacteria no longer dominate the tissue. Their number has fallen below what the damaged tissue can support. Antibodies remain bound to fragments. Complement has been inactivated or cleared. Neutrophil recruitment is slowing. The endothelium is returning toward its ordinary state. The immune response cannot simply continue until every microbial molecule has vanished. Fragments may remain after the living thread is gone. If pattern recognition treated every fragment as a reason to sustain full inflammation, the tissue would never heal. The system must distinguish a declining danger from a continuing invasion. Several changes provide that information. Microbial replication stops. Damage signals decrease. Neutrophils undergo apoptosis instead of spilling their contents. Macrophages engulf them through a quiet cleanup process called efferocytosis. Dying cells expose signals such as phosphatidylserine on their outer membrane, allowing them to be removed while their contents remain enclosed. The act of clearance changes the macrophage. Inflammatory cytokines fall. Specialized pro-resolving mediators limit further cell recruitment, promote debris removal, and support repair. If this clearance fails and dying cells rupture, internal molecules spill into tissue, making persistent inflammation and loss of tolerance more likely. When clearance succeeds, the chemistry of the tissue begins pointing away from attack. Fibroblasts move into the injured region and produce matrix proteins. Small vessels regrow. Keratinocytes advance across the surface. Collagen is deposited and remodeled. If the injury was shallow, the original architecture may be restored with little trace. If it was deeper, a scar may remain. A scar is not a failed repair. It is a rapid structural solution that trades some original organization for strength and closure. The tissue does not return exactly to its earlier state. It becomes functional enough. The immune system also does not return exactly to its earlier state. Most expanded T and B cells disappear. Some plasma cells remain in bone marrow. Some memory cells circulate. Some remain resident near the barrier. Antibody concentration may decline slowly. The repertoire has changed. If the same bacterium enters again, the early response will still involve barrier cells, complement, neutrophils, and macrophages. But antibodies may bind sooner. Memory cells may expand faster. The lymph node will not be starting from the same rare initial clone. The body carries history as cell population and molecular concentration. This memory is useful without being perfect. The organism may change its surface molecules. The next wound may occur in a different tissue. Age and health may have changed. A memory formed in blood may not prevent colonization at a mucosal surface. Protection is a head start, not a guarantee. The completed response can leave subtler changes in stromal cells, vessels, nerves, and resident immune cells. Sometimes this creates faster local readiness. Sometimes it contributes to fibrosis, chronic pain, or recurrent inflammation. The boundary between useful memory and pathology depends on whether the change remains proportional to future need. That balance is never perfect. The immune system is sometimes praised as though it performs flawless recognition every moment. It does not. People develop infections, cancer, allergy, immunodeficiency, transplant rejection, and autoimmune disease. Pathogens escape. Tumors suppress. Tissues scar. Responses arrive too late or persist too long. The remarkable feature is not perfection. It is that a changing organism can maintain workable boundaries for decades while eating foreign molecules, breathing microbes, carrying resident communities, repairing constant microscopic damage, and creating receptors partly at random. Most potentially dangerous encounters end without becoming memorable illness. The skin closes. The lung clears particles. The intestine tolerates dinner. A damaged cell dies without inflammation spreading. A virus-infected cell is removed while its neighbors survive. A self-reactive lymphocyte remains silent. These quiet outcomes are difficult to count because success leaves little evidence. The absence of disease is not the absence of immune activity. It is often the product of well-timed activity and restraint. Right now, the body contains many immune scenes at smaller scales. In bone marrow, stem and progenitor cells are producing new blood cells. In lymph nodes, naive cells are moving across stromal networks, testing antigen-presenting surfaces and finding nothing that requires expansion. In the intestine, immunoglobulin A is entering mucus and coating microbial communities. In the lungs, macrophages are clearing particles carried into alveoli during the day. In skin, resident memory cells remain near old sites of exposure. In the liver and spleen, macrophages are recycling material from aging blood cells. In tissues, regulatory signals are preventing harmless antigens from becoming inflammation. Some cells are dying because their task is complete. Others are dividing because replacement is needed. The immune system is active without being alarmed. There is no task for the conscious mind within this process. No receptor needs to be directed. No lymph node needs to be reminded to filter lymph. No antibody needs to be held in place. The body's defenses are not improved by remaining awake to supervise them. They are part of the same physiology that continues breathing, circulating blood, regulating temperature, and repairing tissue without deliberate control. The phrase immune system can now become less like an army and more like a conversation. Barrier cells speak first. Innate sensors describe broad patterns. Dendritic cells carry context. T cells interpret peptide and provide direction. B cells refine molecular recognition. Antibodies extend that recognition into fluid and mucus. Regulatory cells lower the volume. Macrophages clear what remains. Tissues answer by healing. The conversation is not always calm. It can become loud enough to cause fever, swelling, cough, pain, or exhaustion. It can mistake pollen for danger or self-tissue for a target. It can become too weak to contain infection or too restrained to eliminate cancer. Medicine increasingly works by changing who speaks, which signal is heard, and how long the message continues. The deepest discovery of modern immunology may be that defense and restraint are not opposing systems. They are parts of the same system. A response without restraint becomes disease. Restraint without response permits disease. Health exists in the changing interval between them. The splinter wound is quiet now. The blood vessel no longer displays the same adhesion molecules. The chemical gradient has flattened. A final macrophage moves through repaired tissue and engulfs a remaining fragment. No new neutrophil follows. Farther away, one memory cell remains. It carries a receptor assembled from gene segments before the encounter began. The receptor was selected by a peptide carried from the wound. The cell divided, performed its task, and survived the contraction that removed most of its clone. It does not remember in the way a person remembers a scene. It simply remains changed and ready. That is enough. The body does not need to keep the inflammation in order to keep the lesson. The pain can end. The swelling can fade. The fever can fall. Memory can remain without continuous alarm. Outside awareness, millions of other decisions continue. Most end in tolerance. Some end in repair. A few begin new responses. All are constrained by the same quiet requirement. Protect the organism without destroying the place being protected. For tonight, there is nothing more to follow. The node can return to its ordinary rhythm. The antibodies can circulate without being counted. The resident cells can remain at their surfaces. The immune system can continue its distributed work without becoming the object of attention. The body is not undefended because it is resting. It is resting inside a defense that has never been separate from the body itself. Sleep well. Good night.